Okay, so next we thought we'd show some examples of integrative analysis that has been done in ENCODE and modENCODE consortia and a few other places, just to start to get a feel for the sorts of things you can do when you bring data together generated by many, many different groups, simply on a scale that no one group would achieve. So shown here on this first page is just a sort of overview of some of the stuff that's been done in ENCODE and modENCODE. On top there you see the sort of perfunctory ENCODE genome annotation plot, and then under that we've got a few particular examples. So this first plot down here on the left, which they can't see, can they see me pointing at the screen, or they're only seeing slides? They're only seeing slides. Okay, so they can't see my laser pointer. All right, the next plot. I can do what now? Oh, that works?
Yes.
Oh, great. Right here in this plot, you can see many different chromatin marks, transcription factor binding events laid over the top of genome-wide averages over gene models, and then other annotated events. And it's just a way of starting to visualize the data sort of en masse, and then a few specific examples pulled out. It is just sort of very low-hanging fruit, the kind of stuff that's been done in the past. This sort of very descriptive analysis won't necessarily fly in the future. We'll have to move beyond this. One of the cooler findings from ENCODE was looking at all of the chromatin marks and then asking how they behave with respect to cage peaks and certain transcription factor binding events, and finding that virtually all chromatin marks had asymmetric distributions around particular types of events. And people hadn't seen that previously because they'd done these giant genome-wide averages that were not directionalized with respect to strand. And after you took into account that direction and did some creative co-clustering, you could really see these very anti-symmetric distributions for chromatin marks. So that was pretty cool. They discovered lots of new genes. Here's from modENCODE. In particular, as Karsten and France both alluded, lots of long non-coding RNAs and genes that encode only very short polypeptides. For most of the genes that encode short polypeptides, the information on translational status comes from conservation evidence, which in some cases is extremely compelling, and others less so. And it's still really unclear how much is coding versus non-coding. And in zebrafish, you have a really unique resource of prodigious amounts of ribosome profiling data coming from various community members that might go to answering some of those questions in a more robust way than has been done in other model systems. Some of the analyses on the previous slide were sort of pretty complex overviews of many different data types. So now I'll show you a few much more simplistic and sort of concrete examples. So one of the first things we ever did in ENCODE, very natural thing to do, is predicting gene expression levels from chromatin data. So predicting the intensity of cage peaks, predicting FPKM or RPKM over gene bodies from RNA-seq and things like this. And on the left here on this slide, you see a sort of pipeline for doing this kind of analysis of uniformly processing data, registering it onto the genome, fitting classifiers, fitting regressors, and then predicting quantitative levels of expression. And it's been known for quite a few years now that you can predict steady-state mRNA levels pretty effectively from chromatin data, but the standard deviation on these estimates is still a couple of logs. So although the R-squareds look great, when you get down to the nitty-gritty of how much we really know about steady-state mRNA levels from chromatin and transcription factor alone, it's still actually not that impressive. There's a tremendous amount more to learn. We've also worked on predicting protein levels from subcellular fractionated RNA-seq. This was trying to predict the distribution of 1 million mass spectra from whole genome shotgun tandem mass spectrometry from subcellular fractionated RNA-seq data. The coolest finding from this paper was that the best predictor is the poly-A minus cytoplasmic fraction. So you'd think it'd be poly-A plus cytoplasmic mRNAs, right? The level of mRNA predicts the level of protein. It's not true. The poly-A minus cytoplasmic fraction is a much, much stronger predictor. And that's presumably something to do with the fact that the event of having the degradation products of the mRNA post-translation is a better predictor of the actual event of translation than the mRNAs themselves. And it's just a cool thing you can do when you have a couple data types. Both the last two slides amount to prediction problems where you've got some outcome like protein level or gene expression level that you want to predict as a function of a bunch of inputs. And I imagine that, as Ferenc said, many of the things that we tackle in this consortium are going to boil down to that sort of regression problem, and I'll suggest some examples of that in a few slides. One of my favorite findings was from Snyder's group in the last round of joint ENCODE modENCODE publications. So one of the things he thought he was going to get out of analyzing 3 species where we had many hundreds of transcription factors chip-seqed in each organism was that the co-association profiles for the transcription factors were going to be conserved. They were going to match up. So you look at CTCF, you ask where does it bind, and then you ask which other transcription factors bind with it. And then you ask, is that the same? Is that cohort the same in human, worm, and fly? And the answer was no. In fact, it looked bloody awful. It wasn't the same for a single transcription factor. None of the imputed transcription factor interaction events were conserved. But that's because they were looking genome-wide. They were taking these whole genome averages. You know, genome-wide, does CTCF tend to associate with, you know, SOX2 or whatever? And they got just bubkas. They used this approach illustrated here of trying to fit self-organizing maps to the genomes. And what that does, I mean, don't worry about what self-organizing maps are. What the approach does is it tries and finds subsets of genes. It's basically biclustering. You try and find subsets of genes that are bound by CTCF where the interaction profile is the same in the 3 organisms. And that works blindingly well. And then you see immediately that these subsets are densely conserved across the entire metazoan phylogeny, including human, worm, and fly. But the key thing, the key observation is that these processes are heterogeneous across the genome, and there have been lots of little independent trajectories for particular gene families that are poorly conserved. So you have to pull out individual gene families, individual processes to see the conservation of things like interactions. And data heterogeneity is something that we'll be able to tackle in zebrafish very effectively simply by virtue of having dense time courses and lots of data. That said, the developmental time courses alone are not completely untrodden territory by any means. Shown here is an example of predicting the developmental trajectory of regulatory interactions in a Drosophila developmental time course. So this is looking at ChIP-seq and RNA-seq data across many different developmental time points in fly and asking if you can predict these sorts of branching events in regulatory hierarchies, asking when particular pathways turn on and for which genes they're acting as effectors. In fly, this was very low-hanging fruit. It's beautiful work from Jason Ernst's lab at UCLA. But the time course in fly was very low resolution. It was 2 hours resolution through embryogenesis, and then a couple larval data points, a couple pupal data points, a couple adult data points. And so in that sense, making these sorts of predictions is pretty low-hanging fruit because you're essentially, and further, this is an ecdysozoa, so you're essentially at the level of ecdysone pulses. And getting the secondary interactions of that particularly nuclear or hormone receptor. In fish, we'll have much richer information than this. Be interesting to try analyses of this kind. Another thing they did that is kind of interesting to point out is they tried to predict the go terms of genes from multi-omics data. So they just took all the ChIP-seq data, all the chromatin data, all the RNA-seq data, everything they had, and asked, given a high-dimensional profile for each gene, so something like a 2,000-dimensional profile for each gene or more, can you predict its go term? And for many go terms, the answer was yes. You have to keep in mind that there are a lot of genes, and these processes don't annotate many. So an AUC of 0.94 actually genome-wide isn't that great, but still. There's predictive power for go terms for multi-omics data, but no one yet has provided real insight into why prediction is possible for this problem, or indeed the vast majority of problems that have been tackled in this way. So you know, predicting go terms for highly derived cell lines is one thing, but in zebrafish you have really incisive anatomical and developmental ontologies that might allow a lot more power for this kind of analysis and a hell of a lot more interpretability. Right, a lot more interpretability in saying, okay, we can predict cellular respiration from multi-omics data. Well, that's great. What does that mean? In fish, this sort of analysis might correspond to really getting at the roots of organogenesis, stem cell specification, et cetera. ENCODE also had some important negative findings that are worth pointing out. And this is ongoing into the current consortium. We still can't predict enhancers with sufficient accuracy to enable genome-wide scanning. So people can take balanced training sets of 50% active enhancers, 50% sequences that were tested but inactive, and you can get superb AUC on these balanced training sets in both human and mouse. You know, 0.9 easily, sometimes up to 0.95. But you have to consider, what does 0.9 mean on a balanced training set for when you go genome-wide? So you're going from a marginal distribution of 50-50 to a distribution of a marginal distribution of less than 1%, right? If you take genomic sequences at random, probably 1%, less than 1%, are going to function as enhancer elements in these assays. And that's a classic statistical problem called mismatch marginals. And so actually in ENCODE, if you look at the elements that they've annotated as enhancers in K562, for instance, Brock Cohen had a beautiful paper last year where he showed that they'd achieved about 23% positive predictive power. And so that's the state of the art, that if you test elements from a genome-wide scan, even at the top of the rank list, 80 to 90% are going to be wrong. They're going to be false positives. So there's still a major false positive problem in the prediction of functional elements, and that is ongoing in the current ENCODE consortium. It's the focus of like 60% of our teleconferences, in fact. We can do better in some tissues than others, and we don't understand why. Forebrain enhancers in mouse, for instance, at embryonic day 11.5 are way, way easier to predict than heart enhancers. We suck at heart enhancers. However, if you threshold on methylation status in any tissue, and especially in heart, we can predict hypomethylated elements. So you have a genomic sequence. You tell me whether it's hypo or hypermethylated. I'm going to do a lot better job for the hypomethylated at telling you whether or not they're going to be an enhancer than I will for the hypermethylated. Marks like H3K27 acetylation lose a lot of power on hypermethylated elements. There's shown here a giant network hairball. ENCODE, modENCODE, and many others have been subjecting the genomics community to these beasts for nearly a decade. We still gain no predictive power by trying to regress on networks instead of the underlying raw data. So we haven't yet gotten to a point where these sorts of models become really useful and improve our functional understanding of underlying biology. And as Ferenc said, if we can use dense time courses to get to bona fide causal relationships, we might end up with something that's a hell of a lot more informative than a hairball. RNA chromatin interactions. I want to call out this particular area of inquiry because I think it's a frontier and I think it's one we can approach. Luco had this beautiful paper in 2010 in Science where she showed that you could predict splicing status pretty well from chromatin marks, and she identified which chromatin marks were important and where they needed to fall over exon-intron boundaries. This really built, if you look, what's the history of this work, where did it come from? This built on Julie Ehringer's beautiful work in C. elegans, where she discovered that H3K36ME3 maintains the maternal memory of splicing events in the embryo. So it paints constitutively spliced exons, and the quantitative level of H3K36ME3 over cassette exons is predictive of their level of inclusion. Luco's now found long non-coding RNA, and so have a few others, implicated in transducing chromatin RNA interactions during splicing. But so far, all the new examples of this are in cis. So of course, right, the spliceosome has RNA components, lots of other things have RNA components. We know there are RNA components in transducing and executing splicing. But it'd be nice to find some new fundamental components of ribonuclear complexes, and we haven't done that in other systems yet. So we've done some work within ENCODE too on predicting splicing status from chromatin. Been working with Roderick Guigo's group for years on this. The alternative splicing can be predicted in the human cell lines from chromatin data with some sort of vague accuracy, still not up to 0.8 AUC. When you do this, you recover the well-known central importance of H3K36ME3, and you get this sort of hub-like structure I'm showing here. This is a depiction of how our various chromatin-based predictors are interrelated. But you see that H3K36ME3 is not acting alone. It's interdependent with lots of other chromatin marks, and most of those interdependencies are nonlinear. Here's a visualization of some of those interdependencies, where on the x-axis we have the quantitative level from ChIP-seq experiments of various chromatin marks, and then on the y-axis you have the likelihood that a given exon will be included. And you see these sorts of very interesting sloping sort of and-like features for the rules that are represented by the levels of these chromatin marks as measured by ChIP-seq data. And we don't understand why these patterns exist. No one knows what is transducing this information, why you have nonlinear interactions between all these chromatin marks. We detect nonlinear interactions of up to order 7. I mean, that's an incredible complexity in the combinatorial code of splicing that's only been touched. When you include transcription factors along with histones, you don't do much better than just histones alone. When you include RNA binding proteins, particularly members of the spliceosome, you can predict splicing with almost deterministic accuracy. And what's interesting, if you look at the interaction rules and the form of the interaction between, between RNA binding factors and then look as a function of the level of RNA binding factor as detected by eClip. Look at the percent exon inclusion, or the likelihood that a given exon will be included in a mature transcript. You see that the rule has an OR-like structure. If either of these RNA binding factors is well bound, that the exon is going to be included, and if neither, it won't be. That's in contrast to the sorts of rules that we see for transcription factors regulating transcription, where we get these AND-like rules, where we require 2 or more factors to be present before we see transcriptional activation. That's probably just an artifact of the factors that have been tested in these experiments, but it's a cool potential direction. It's all completely unpublished stuff. RNA chromatin, so more about RNA chromatin interactions. So Luco and Sell in 2011 had this nice little review. The molecular basis for RNA chromatin interactions is really clear. Something that's been missing from this entire story and is still missing is the role of 3D chromatin structure in assessing these interactions. Since people first started doing chromatin structural assays, we've thought a lot about transcription factories and stuff like this, but what about the effects on co-transcriptional splicing? How does 3D chromatin structure localize and locally increase the effective concentration of RNA binding proteins? How does that relate to the determination of the information that will ultimately be encoded in mature RNAs? Further, and very little is known about the specific molecular players. In fact, you see this little diagram from 2011, very little has changed. We still know only a few of the specific proteins that are involved in transducing information encoded in post-translational modifications of histones to mature transcripts. And note that some of these bubbles are labeled very nebulously, right? This one says HNRNPs. Well, that's great. Heteronuclear ribonuclear proteins. That's informative. So there's a tremendous amount that is completely unknown here that we may be able to impute from some of the data to which we'll have access. Less, even as little is known here, much less is known about the transduction of chromatin information to translational regulation. We can predict levels of translation from chromatin data, not super well, but we can. Why is that, right? I mean, there's a few lysine demethylases that have been shown to interact with the ribosome, but there's a big frontier there. Very little is known. So here on the next slide, I've just tried to summarize some of these emerging directions for multi-omics work. So the RNA chromatin interaction stuff I've harped on a bit. I think we have an opportunity to learn more about promoter selection. There's a lot of exquisite cage data in zebrafish, and that cage data has been collected in the context of well-defined developmental periods where we also have ChIP-seq and many other data types, DNA methylation, chromatin structure. There's a lot more we may be able to say about how promoters are actually selected, and possibly even with temporal resolution, which would be fantastic. One question I have for the community actually is if anyone is collecting or is planning to collect BRU-chase data using the BRU-seq platform. So trying to collect dynamic information about the rates of splicing and transcript elongation. Splice site selection and splicing efficiency. We should be able to say a lot about that. Integrating the methylation 4C chip and RNA-seq data should be very powerful for learning something new about that. Polyadenylation site selection. There's been some work in this area, but new stuff's discovered every couple of months. I mean, you know, the ELAV and then the MSI and the quaking-related factors and their role in splice site selection. Lots of brand new stuff all the time. Or sorry, polyadenylation site selection. Co-transcriptional formation of mRNA-bound ribonucleoproteins. As far as I know, we don't have any RIP or eClip data, but if that comes in, we might really be able to make some progress there. In absence of that, we can still work with motif level information. Because remember, we have motifs in vertebrates for over half the RNA, the known RNA binding proteins. So we do know a lot about the sequence specificities of these factors. It'd also be nice to look at, and of course, generally, why can we predict splicing, localization, translation, et cetera, from chromatin, getting down to the nitty-gritty and trying to build some engineering level models of why these processes happen, right? The fact that we can predict is old news. We've got to get to why we can predict. And part of that in zebrafish may be doing things like looking at existing mutants and seeing if we can explain some phenotypes that we've not previously been able to explain. I'll say more about that in a moment. And then when does chromatin first become important for predicting things like splicing, polydenylation, site selection, translation? It's not instant, right? I mean, initially these things are maternal contributions, and then is it the very second you start zygotic transcription, or does it happen much later? My guess is, based on Drosophila work, that it happens much later, because initially the chromatin state in the zygotically transcribed embryo is extremely naive. In fly, it's overwhelmingly H3, K18 acetylation everywhere. And that is not true of any other developmental period. So when does that first start, and can we get a window on it? So predicting active translation from chromatin and splicing information, how much about translational regulation is encoded in chromatin state and structure? Can we postulate some of the factors involved in these processes? Maybe, that would be great. Imaging, Boris is going to talk about imaging data to disaggregate multicellular and whole organism sequencing data to try and get some spatially resolved atlases in high throughput, much higher throughput than is allowed by the two photon approaches. And then identification of the drivers of organogenesis. Can we predict early expressed genes in primordia using ChIP-seq and or RNA-seq? We've done some work on this in fly. You absolutely can. It works great. Boris will tell you about some more in zebrafish. Integration of single-cell and multi-omics data. The DANIO-CODE ITN is going to likely produce some single-cell data, so we should start thinking about how we're going to integrate the single and the more standard approaches. Quantitative models of complex tissues can be constrained by single-cell measurements, and we can try and start to impute some dynamics. If anyone's doing live single-cell, live single-molecule imaging in zebrafish, that would be really spectacular for that sort of biological question as a mode of validation and model constraint. We can impute function for cryptic genes. Guilt by association studies work really well, especially in multi-species analysis. We're not collecting, zebrafish does not exist in isolation. We know a lot about human. Karsten showed you a great slide from ENCODE, the massive data that's been collected. We've got the modENCODE project, many other organisms with lots of omics data, so we can generate multi-species models. And there are some really nice new techniques for doing simultaneous imputation across many organisms with multi-omics data, where you have loose measures of orthology, and you allow orthology to guide, but not completely constrain imputation for the functions of novel genes. So I think we could go after long non-coding RNA and short ORFs in a really serious way that might be really nicely complementary to efforts that will be going on in Phantom 6 and a few other consortia around the world. We can also really go after explanatory power for human disease, and that's integrated with the work in the Undiagnosed Disease Network in zebrafish. So 96% of disease-related GWAS variants in human are still in non-coding variants and still completely unexplained. There's a whopping 71 long non-coding RNAs that are peppered by GWAS hits for multiple diseases, so you can really be pretty confident those are disease-related long non-coding RNAs. But that's 71 out of 15,000. We have absolutely no idea what the vast majority of these genes do, so guilt by association analysis would really be a hope for that. And so lastly, just pointing to some low-hanging fruit, and this is something we should all brainstorm about collectively. Everyone should write in ideas. It's really obvious that predicting epistatic networks involved in cellular processes where one or more players has a molecularly defined mutant, that's a really straightforward thing to do. Half the genes in zebrafish have been knocked out, so we should be able to make some headway there. We can try and explain the basis of phenotypes where we have a phenotype but we don't know why, concentrating on phenotypes with clear orthologs to human to complement the undiagnosed disease network. particularly leveraging anatomical ontologies. There's a very nice anatomical ontology now linking fish to human. We should leverage it. We could also consider in the ZenCode ITN and with other collaborators trying to make some double and triple mutants to test hypotheses about epistatic interactions. In these networks, we're going to have really complicated, you know, 7-fold, 8-fold epistatic predictions, and going after some of those would be great. We should definitely, as Karsten said, we should definitely try to identify new genes involved in organogenesis and cell fate specification. Conserved protein domains are powerful, but they're not omniscient. Most people are not aware that there are 200 ultra-conserved short ORF genes with ORFs between 11 and 80 amino acids that are, or maybe it's 80 and 100, or 11 and 100 rather, something like that. But really short ORFs where we know absolutely nothing about what they do. Some of them have conserved domain annotations, many don't. But the conserved domain annotations are completely uninformative. The vast majority of them are DUF, domain of unknown function. The ones that have names are just disease association. So BC10 is bladder cancer associated 10. Thanks, that's great. It's knocked out in bladder cancer. We know nothing else about it. And there's things knocked out in 90% of bladder cancers, but nothing else is known. It's conserved across all metazoans, you know, from honeybee to vertebrates, and the zebrafish one looks on. I mean, yeah, it's amazing how conserved these things are, and we don't know a single thing about them. It would be great to really try and use the multi-omics data in fish to go after some of the functions of these. You know, we've tried to do this in other systems and haven't had a ton of success yet. And that's it. Okay, discussion. I'm late. Any questions? Any answers? To include what?
A person called Manuel Irimia, who has lots of splicing data in zebrafish. It was a comment.
Oh, that's great.
So particularly your story on splicing. And also comment that you really have quite a lot of data in DANIO-CODE.
See, that's what they resay.
Yeah, there's lots of data in the submitted material which you can mine for the splicing and when it really starts in development. As far as we can tell, it starts before the main activation of the zygotic genome, already together with the minor activation, but it's very limited at that time. And there's lots of time points to interrogate how it evolves in that period. And using SNP analysis, you will be able to dissect maternal spliced RNAs from the de novo. So I think there's lots of exciting opportunities for the kind of analysis you suggested, also in relation to chromatin and features, which, as you suggest, may predict splicing events. And yeah, there are lots of observations already from zebrafish that indeed these chromatin features are matching with not just splicing, but also other post-transcriptional regulatory events, such as Drosha cut of microRNAs.